Okay. So before we get started, I did want to talk to you all about the mastering A&P assignment. I got several emails about that. I got back to some of them, but I'm sure some of them slipped through the cracks, and I'm sure some of you just hadn't gotten to emailing me about that yet. That assignment should be visible now, so thank you for letting me know that it wasn't. That said, I'm pushing the deadline for the endocrine stuff until the end of next week. I think we're all feeling a little burned out right now, though I hope you all got some sleep last night. So don't worry about that. If it is not visible now, please do let me know, because now I double checked in multiple places and it should be. So if it's still not there, I'll let me know. But otherwise, just try to get to that sometime by the end of next week. Okay, one more person filing in. Okay. So our job for today is to get through the somatic nervous system. And we're going to do a little bit of the beginning of endocrine. Endocrine is complex, and we're going to think about the system in kind of two ways, both in like a functional way and in more of an anatomical way. So we're going to do a little brief intro to that at the end of today, but we'll save most of that for Friday and possibly Monday. Okay. But somatic nervous system is hopefully pretty straightforward. We'll be coming back to this next week as we start to talk about skeletal muscle and contractions. So for right now, we're really just thinking about the nervous component. We're going to come back to how the discrete muscle cells work very soon. Okay. So I am sharing my screen. Cool. Okay. So we're just trying to figure out right now the functional anatomy of the somatic nervous system, the neuromuscular junction. So we're trying to figure out what these nerves look like and how they join up to our muscles. So we're trying to figure out what's going on with that nervous connection. And we like to understand a little bit more in depth how that junction between the nerve and the muscle works, so that we can understand some weird things that happen there. So there are a couple of poisons that target specifically the neuromuscular junction. So the joining between the somatic nerve and the muscle cells it targets. And this is pretty interesting and relevant because it can kind of freeze up your skeletal muscles, right? Really paralyze you. And it's all down to actually how that junction works. So we'll take a look at that as well. Okay. So I'm pulling us back first to our big map of the whole nervous system. So remember, we had the central nervous system that's really for information processing, which we refer to more scientifically as integration. So that receives sensory information and sends out motor commands. So for this unit, we've been talking about the different parts of the motor division. So the efferent going out, motor division of the nervous system. So the motor division is going out, forming the peripheral nervous system, so everything other than the brain and spinal cord. And it splits into this first division, somatic versus autonomic. So we've been talking about autonomics. We've been talking about parasympathetics, our rest and digest, our sympathetics, our fight or flight. It's important to note here that when we were talking about those autonomics, we were talking specifically about nerves that do unconscious processes, go to smooth muscle cell that we'll see in like in blood vessels, cardiac muscle in the heart, glands, releasing hormones, all this unconscious stuff. So the somatic nervous system is a whole different beast. The somatic nervous system is going to skeletal muscle. So the somatic nervous system is going to be sending nervous impulses to muscles that are under your voluntary control. So that's the big division between somatic and autonomic. That somatic is going to be voluntary in large part. So somatic nervous system to skeletal muscle. Okay. So when we're thinking about the somatic nervous system, we got motor neurons coming out of the central nervous system into the peripheral nervous system. These motor neurons, just like our like all motor neurons, are going to originate in the ventral horn. Okay. Because we're talking about the somatic nervous system, the place they're going is skeletal muscle. That's a definitional thing for the somatic nervous system. The neurotransmitter that these neurons are going to release is acetylcholine. So last time when we met on Monday, we talked a lot about the different neurotransmitters involved in the autonomic nervous system. We looked at acetylcholine, we looked at norepinephrine and epinephrine. When we're thinking about the somatic nervous system, easier, always acetylcholine. So somatic acetylcholine. Cool. So when in doubt, honestly, pick acetylcholine. It is the more common neurotransmitter involved in these systems. Okay. So our receptors in skeletal muscle, we're going to have cholinergic receptors because cholinergic receptors receive acetylcholine. Okay, so if our nerves are giving off acetylcholine, we have to receive acetylcholine. Specifically, our receptors in skeletal muscle are going to be nicotinic cholinergic receptors. So we remember that those are basically the ones that open ion channels. They're fast. They're a version of an ionotropic receptor. We also saw them in our ganglia, right? When we were having our presynaptic and postsynaptic neurons in the autonomic nervous system, we saw nicotinic cholinergic receptors there. We're seeing them again now in our skeletal muscle. Yeah. Okay, so a bunch of this should be familiar to about half of you, maybe more. So a bunch of the somatic nervous system stuff is really what we've been talking about and looking at in lab this unit, right? So as we've been looking at muscle contraction in lab, right? Our electromyograms, we've looked at the pathway of a motor nerve. Specifically, we were looking at the pathway of a somatic nerve, right? A somatic nerve in the efferent division of our motor system. So we saw that it starts from the ventral horn. So we're continuing, forming a spinal nerve, right? Our ventral root going into the spinal nerve. Then that motor neuron is going to go out. It's going to find itself a skeletal muscle. So those are like the named muscles that we're doing in anatomy lab this unit. If we zoom in a little deeper, we know that if we track a single motor neuron, it does this cool thing where as it approaches its skeletal muscle that it's innervating, we're going to see that it splits. So our single motor neuron is going to have many, many different endpoints, each of which will go to a muscle fiber. And if we look at any muscle fiber, we can see that we can trace it back to a specific motor neuron. Okay, so the fact that we have one motor neuron for many muscle fibers, but each muscle fiber goes back to only one motor neuron is important. So just kind of try to keep that relationship in your mind. One motor neuron, many muscle fibers, but any individual muscle fiber will have one motor neuron assigned to it, because this is what's going to form our motor units, right? So one motor neuron as it splits into many muscle fibers. If we want to talk about all those muscle fibers together that are innervated by that one motor neuron, we're calling them a motor unit. This is important because they will largely act together. Okay. So our somatic motor neurons are innervating the skeletal muscle in groups in motor units. Okay. So each muscle fiber only has one motor neuron assigned to it. We don't have one muscle fiber that's both red and blue, right? They're going back to one specific motor neuron. Okay. So now we're going to look at the junction between the ends of those motor neurons and those muscle fibers. So down here, for example, right? So we're looking at what we call the neuromuscular junction. So the neuromuscular junction is where we have the connection, right? Synapse basically of our neuron and our muscle fiber, which is our muscle cell. Okay. So we're going to start with what's going on the motor neuron side. So on the motor neuron, right, if we went way back to our beginning, to our understanding of how neurons work, right, the thing that would happen first because we have to figure out whether that motor neuron is going to fire, right? So whether that motor neuron gives off a signal, whether it tells our muscle to contract. So the activation of our motor neuron is going to be dependent on the summation of all sorts of EPSPs and IPSPs, right? So those postsynaptic potentials, so it's receiving signals from other neurons all around somewhere in the central nervous system. We have those integrating centers. So they're creating these graded potentials in the cell body of our motor neuron. We're adding them together, maybe subtracting them if we have some inhibitory ones. We're trying to figure out whether we get to threshold at the axon hillock, because that's what determines whether this motor neuron fires an action potential out to the skeletal muscle at all. Okay. So that's what's happening with our motor neuron. If it does fire, just like in all our other neurons, we see an action potential travel down the axon, right? So it propagates. We think of it as a wave of depolarization. It would travel all the way down the axon, and we'd see that the axon splits, right? Then we're going to come into our neuromuscular junctions, and we're going to zoom in on one of those junctions in a second. But what the motor neuron is going to do, its job at that neuromuscular junction, is going to be to release acetylcholine. On the muscle side, we then have to receive that acetylcholine, right? So our neuromuscular junction overall is the synapse between this motor nerve, which we've just had this motor neuron fire off an action potential, release acetylcholine. So we're then going to have a synapse with our muscle fiber at that spot where we're releasing the acetylcholine. We do have specialized words for the parts of both the neuron and the muscle fiber in this neuromuscular junction. So in structure, they're really similar to what we saw in our normal synapses in the central nervous system in the brain when we were going neuron to neuron. But now that we're in a connection between specifically a motor neuron innervating somatic muscle, we name things a little differently. So what we previously thought of as our axon terminals, right? Gonna scroll back again, right? So we might have thought of these different endpoints as our axon terminals here. We are now going to call them terminal boutons. I want you to think in your mind right now. So terminal bouton is what we're calling the axon terminal, the ending where we're releasing neurotransmitter for our somatic motor neurons. Part of the reason we want to have a special word here is we had a sort of special and different situation in our autonomic motor neurons, right? So when we're thinking about our sympathetic nervous system, for example, innervating smooth muscle, we saw that we also had a specialized structure there. Okay. So we saw that in our autonomic nervous system, we have these structures called varicosities. So we want to make sure that we know varicosities are for the autonomic. Our terminal boutons are here for our neuromuscular junctions for our somatic nervous system. Okay. So abutting our terminal bouton, right? So terminal bouton is the nerve side, the neuron side. Our motor end plate is the portion of the muscle cell that's involved in this junction. So we're going to have a specialized membrane on part of this muscle cell, on part of this muscle fiber that participates in this junction in this synapse. Okay. So all of our motor neurons coming in are releasing acetylcholine, which means that we have cholinergic receptors on the motor end plate, so on the muscle side. Specifically, they're nicotinic cholinergic receptors. So we can see a picture here. So nicotinic cholinergic receptor happens to also be a type of ionotropic receptor, which means that all it does is it opens an ion channel, right? So acetylcholine binds on, so that would be this little guitar pick thing here, opens up our channel, allowing ions to flow through. When we're thinking about neuromuscular junctions, in the past, when we've seen neurotransmitters binding to receptors, we've talked about both excitatory and inhibitory effects, because when we're in the central nervous system and we're talking about neurons talking to each other, That's true. They can both inhibit or excite. When we're talking about neurons connecting up to skeletal muscle, so the neuromuscular junction specifically, we're going to see that these synapses are all excitatory. So really the only thing a motor neuron can do as it sends a signal out to skeletal muscle is it can create depolarization, right? So it can create an action potential in the muscle cell and therefore create contraction, but there actually aren't any inhibitory signals there. We just like turn off the signal overall and just not have that motor neuron fire at all. So it's important to note, because these synapses are all excitatory, what we would in our neuron to neuron connections call a postsynaptic potential, we're going to specifically refer to as our end plate potential, because we're now on the muscle fiber. So on the motor end plate, we'll have an end plate potential. It's always going to be excitatory. So we don't have to do EPSP and IPSP when we're in our muscle cells. The EPP, always excitatory. Okay. We're also going to see an enzyme called acetylcholinesterase involved in these neuromuscular junctions, which is going to be working to kind of to clear our acetylcholine. My favorite is when they have a nice big picture with everything numbered for us. So this is basically what I've just been saying, but I'll list it out in a nice flow. So let's take a look. So here we see our neuron, our motor neuron, our somatic motor neuron. You see its terminal bouton, right? So its axon terminal is now called a terminal bouton because we're innervating somatic muscle. We're joining up into this neuromuscular junction with a muscle fiber. Okay. So our wave of depolarization, that action potential, gets to the terminal bouton. So just like in our synapses, this opens our voltage-gated calcium channels. So we can see this on the insert. So when the terminal bouton depolarizes, our voltage-gated calcium channels open. When they open, this causes calcium to flow in. Calcium wants to go into the cell down its electrochemical gradient. Okay, so calcium goes into the cell. This causes those synaptic vesicles holding our neurotransmitter, which in this case is acetylcholine, to bind on to the membrane, which then breaks open to release the neurotransmitter. This process is called exocytosis, right? So we can see exocytosis happening here. Now we have our acetylcholine out in this cleft, right? So what we would previously have called our synaptic cleft here. Now in our neuromuscular junction, we have a similar kind of space, like a nice tight space. We didn't see that in our autonomics, specifically in our sympathetic autonomics, but we do see that here in the neuromuscular junction. Okay, so we then bind this neurotransmitter on to our nicotinic cholinergic receptor, which is a channel, opens in response to the binding of acetylcholine. When it opens, it does allow sodium and potassium to flow through. They flow in opposite directions, but the sodium gradient is always really big. So sodium wins. So this is going to depolarize and therefore excite our motor end plate, which is this muscle fiber part of our neuromuscular junction. So we have a net movement of positive charges in, causing depolarization, which we would call our end plate potential. So that's the same thing, just this getting more positive because sodium is coming in. It's an excitatory end plate potential. Okay. The end plate potential is then going to spread out. And it's going to cause an action potential, so depolarization in the rest of the cell as well. And yeah, this happens more easily than when we were talking about graded potentials adding together in neurons. Just so you know, the action potentials are much easier to fire off in the muscle cell. So the end plate potential is going to cause an action potential pretty much. Finally, we sweep up our acetylcholine. The way we do that is that some of our acetylcholine is going to bind to acetylcholinesterase, which is an enzyme on the motor end plate, so on the muscle fiber side here. So when some neurotransmitter hits our enzyme instead of our receptor, whoop, acetylcholinesterase breaks it down. The choline part is going to get taken back up by our motor neuron. So it's going to go back into our motor neuron, which is then going to use it to make more acetylcholine. Questions about this process? This is hopefully very, very familiar because this really is just like our other synapses, just with some new terms. So new words for hopefully very familiar ideas. And just very specific words now that we're talking specifically about a nerve joining up to a muscle. Let's go ahead and take a look at this. I'm going to have to press a couple extra buttons, so give me a second.
Yep.
Nerve impulses, also known as action potentials.
Can you guys hear the sound? I'm gonna
Travel from the brain or spinal cord to trigger the contraction of skeletal muscles. An action potential propagates down a motor neuron to a skeletal muscle fiber. The site where a motor neuron excites a skeletal muscle fiber is called the neuromuscular junction. This junction is a chemical synapse consisting of the points of contact between the axon terminals of a motor neuron and the motor end plate of a skeletal muscle fiber. The events at the neuromuscular junction occur in 7 coordinated steps. Step 1. An action potential travels the length of the axon of a motor neuron to an axon terminal. Step 2. Voltage-gated calcium channels open and calcium ions diffuse into the terminal. Step 3. Calcium entry causes synaptic vesicles to release acetylcholine via exocytosis. Step 4. Acetylcholine diffuses across the synaptic cleft and binds to acetylcholine receptors, which contain ligand-gated cation channels. Step 5. These ligand-gated cation channels open. Step 6. Sodium ions, shown here in red, enter the muscle fiber, and potassium ions, shown here in blue, exit the muscle fiber. The greater inward flux of sodium ions relative to the outward flux of potassium ions causes the membrane potential to become less negative. Step 7. Once the membrane potential reaches a threshold value, an action potential propagates along the sarcolemma. Neural transmission to a muscle fiber ceases when acetylcholine is removed from the synaptic cleft. This removal occurs in 2 ways. 1. Acetylcholine diffuses away from the synapse. 2. Acetylcholine is broken down by the enzyme acetylcholinesterase to acetic acid and choline. Choline is then transported into the axon terminal for the resynthesis of acetylcholine.
without further ado, I'll hand over to our presenter, Hayfen, to talk you through RStudio.
Cool, thank you. Thank you for coming, guys. So everyone has got an RStudio installed on the computer. So if you want to open RStudio, I think the way that, I think the best way for this workshop to work is basically, as long as you can all see.
That is coming from me. All right. So scintillating as usual. Okay. So here we can see a comparison just of some of the key properties in the autonomic and somatic nervous systems for your comparison later. So good for studying. So we want to think about the neurons in our pathway. So we saw when we were thinking about our parasympathetics and sympathetics, we had those ganglia we were going to. We were talking preganglionic, we were talking postganglionic. Our somatic nervous system is pretty simple. We just have the one motor neuron going from the central nervous system out to our skeletal muscle, which brings us to this next difference, right? Skeletal muscle is our target for the somatic nervous system. Our effector organs for parasympathetics and autonomics, our cardiac muscle, smooth muscle, various types of glands. In the case of sympathetics, including adipose tissue, which we saw was often an exception to the rule in our sympathetics. Okay. So the neurotransmitters occurring at the neuroeffector junction. So when we say neuroeffector junction, so in that case, for our somatic nervous system, we've just been talking about the neuromuscular junction between the nerve and the muscle. So the neuroeffector junction for the parasympathetics and the sympathetics would be between like our postganglionic cell and cardiac muscle, for example, would be a neuroeffector junction for the parasympathetics here. Okay, so at that neuroeffector junction, in our parasympathetics, we're going to see acetylcholine, just like for our somatics. And then our sympathetics would be the odd man out, because we're going to see mostly norepinephrine. Okay, so we can then think about our receptor types. We've just been talking about basically ligand-gated ion channels in the neuromuscular junction. So those were nicotinic cholinergic receptors. Similar to what we see in the parasympathetic system, but not quite. The parasympathetic nervous system, we saw muscarinic cholinergic receptors. The difference here is that those muscarinic cholinergic receptors involve a G protein, so that would be a metabotropic response. So that's not always going to be excitatory. It's either excitatory or inhibitory. Whereas our somatic nervous system, we really always have an excitatory end plate potential. And then we saw our many different types of adrenergic receptors for the sympathetics. Okay. And we've talked about these as well in both our parasympathetics and sympathetics. Sometimes we'll see excitatory effects, sometimes we'll see inhibitory effects. Somatic, it's always excitation. A nervous impulse is always like, okay, let's contract this muscle. We can do that voluntarily in the somatic nervous system, not so for our autonomic nervous system, either type. Okay. So you should be able to look back at this table and answer all sorts of questions for yourself. And I highly recommend using tables like this to create practice questions for yourself, because being able to tell these things apart is really important to understanding how the nervous system works. So I said we were going to talk for a second about toxins. So toxins are cool but scary, right? So toxins that can paralyze you work in some different ways, targeting different parts of this system. So we have 3 toxins here that have effects on the neuromuscular junction. So we have latroxin and crotroxin, which are kind of like opposites of each other. And then we have curare, which is a poison from a poison dart frog, which works in a 3rd way that ends up with similar effects. Okay, so all of these can cause something kind of like paralysis, but like it'll be paralysis of different kinds, I would say. So we're going to take a look. So the first one we're going to think about is latroxin. So what latroxin does, I've put a green circle here around number 3. So what latroxin does is basically cause the release of a lot more acetylcholine, lots and lots more acetylcholine. So it basically ends up overwhelming the muscle. Because when we have lots and lots of acetylcholine, we're hitting lots and lots of receptors in the muscle. We're allowing a lot of depolarization over and over again in that muscle. So basically we're going to cause like spasms in the muscle, which can lead to the point of rigidity when this muscle is totally locked up, totally overwhelmed, totally stuck in this kind of contracted state. Okay. So that's one thing we can have go wrong with this system. The opposite of this, so the way we would carry this, crotroxin. So crotroxin has the opposite effect. It inhibits this release of acetylcholine. So the way in which this causes problems for your muscle cells is that when we don't have acetylcholine, we don't open this receptor. This then means we like can't contract that muscle. So when we can't contract the muscle at all, right, like we're going to feel like we can't control our body. So we're going to feel paralyzed because of this flaccid state of the muscles, basically. So they're not going to be stuck, crunched up. You know, they're going to feel flaccid, but because we can't control them, we're in something called flaccid paralysis. Our final toxin, curare, works on the muscle side rather than on the neuron side. So both latroxin and crotroxin were affecting the release of acetylcholine. Curare acts on basically our receptors on the muscle side that usually receive acetylcholine. So when it knocks those out, no matter how much acetylcholine we have, we basically can't get that signal into our motor neuron. So the effects of this are going to be similar to the effects of one of these, right? You think this is going to be more similar in effect on the muscles to latroxin or crotroxin? Are we going to see like spasms, or are we going to see sort of a flaccid paralysis state if we knock out these receptors? What do you think?
Paralysis state?
Yeah, exactly. Yeah, so we have the same problem where we can't receive the signal, so we end up in the same kind of flaccid paralysis state. And when this affects, for example, right, your lungs, this can lead to death, right? If you can't expand your lungs, then you can't breathe. Okay. So, final thing we're going to get to a little bit about the endocrine system. I want to get through the names of some of these glands basically, and one of the big conceptual ideas about how endocrine glands work, so that we can get into kind of the nitty-gritty of some specifics when we meet again on Friday, because there are a lot of different words for different types of hormones in this unit, and having to do both the conceptual part and the let's talk about every individual hormone part at once is rough. Okay. So we're just gonna talk about some of the basic systems that we're going to be working with as we talk about endocrine glands. So the first thing we want to know is like, what is an endocrine gland? And I don't know how much you remember talking about signaling in your previous classes, but you may remember that the word endocrine actually refers to a type of signal sent to other cells in the body, and specifically a signal sent to the other cells in the body via the blood. So that is what an endocrine gland is. An endocrine gland is some tissue that releases molecules into the blood whose goal is to signal to other groups of tissue. So specifically, the molecules that we're releasing are hormones. So endocrine glands release hormones into the blood with the purpose of affecting other organ systems, other tissues farther away from where that original signal is. The glands themselves, if we look at their development, they're derived from epithelial tissue. So if you were to look at these cells on a slide, like we did in our histology unit, they might look something like epithelial cells. And we're going to group our endocrine glands into two types, our primary endocrine organs and our secondary endocrine organs. When we're talking about endocrine glands, we're going to more associate those with the first type. And the difference between primary and secondary endocrine organs is that a primary endocrine organ, that's the job of that structure. So a primary endocrine organ exists in order to release hormones, and that's the function of it in the body. Whereas we'll see with the secondary endocrine organs that they have another potentially more important job, but also release hormones as like a secondary effect. So for example, we'll see that the heart actually releases some hormones. So that would be an example of a secondary endocrine organ, right? The heart's big job is to pump blood. You wouldn't think of it as mainly an endocrine organ, but we will see that it does release hormones that signal to other parts of the body. So it does have endocrine function. So we'd think of it as a secondary endocrine organ. That's just the division here. Primary, that's its only job. Secondary, it's like kind of an add-on job. So I pulled out this picture, and there's this other picture from your book discussing primary and secondary endocrine organs. I just thought this one wrote it out a little bigger, therefore a little better. So we can see all the places that we're going to have endocrine function. So we can see the ones that we have a lot of glands here in purple, that those are our primary endocrine organs. Okay, so we have pituitary gland, which we're going to talk extensively about. Okay. Hypothalamus, pineal gland, so we have structures up in the head and in the brain. We've got a lot of stuff in the neck. We have glands in the neck that release a lot of hormones. The thyroid is a really important one. It's one they like to test with lab tests a lot at the doctors, right? As well as our parathyroid glands, also going to be primary endocrine organs. We've already talked about our adrenal glands. Now we can see them here. So our adrenal glands, also called our suprarenal glands, they sit on top of the kidneys. So they're down in the abdomen. They release various hormones. We've talked about the medulla and how it releases epinephrine and norepinephrine. We'll talk in the future about the cortex, which is like the shell of the gland. It's going to be releasing cortisol and other things, so more stress hormones. We'll see the pancreas, really important for diabetes, right? The pancreas is an endocrine organ. It's a primary endocrine organ. That's its main job, is to release hormones. Yeah, and then we see organs with secondary endocrine functions. So organs that do something else, but also release hormones. We have things like the heart. So heart is a really big job pumping blood. You got the thymus. It sits kind of above the heart. It has some immune functions. It's mainly only seen in children, really. I don't even know if we'll see one in lab. Sometimes in lab we see a good one, but relatively rarely. Adipose tissue, right? Mostly for holding energy reserves, but also has endocrine function. So these would all be examples of secondary endocrine organs. Digestive tract, kidneys, gonads, ovaries. We got lots of secondary endocrine organs. I got them written out here. So the gonads, depending on what parts of them we're talking about, could be considered a primary endocrine organ. Okay, yeah, this is just a list, and I went through the list, but I do want to talk for a second here about some of the hormones that we'll see in the future involved with our secondary endocrine organs. We won't really get specific ones we're looking at here, I think, until AMP2. So we'll be looking at atrial natriuretic peptide and erythropoietin when we start talking about the composition of blood, really. So we'll be talking about those later, secreted by the heart and kidneys. The GI tract has several hormones that it releases that control hunger, kind of, which is pretty cool. And we'll talk about insulin in depth. That goes with our pancreas, but we'll also see some insulin-like growth factors involved with the liver. We're probably not going to talk about leptin or D3, but there are a lot of important hormones released by organs that we don't necessarily think of as endocrine organs. So the really big conceptual thing that I wanted us to get to was to talk about the relationship of the hypothalamus and the pituitary gland. Because the way that the structure works and the ways in which they're connected lead to some kind of complex feedback loops we're going to see between the specific hormones that we are going to talk about by name in detail. Okay. So just a reminder, hypothalamus, part of our brain. All right, we see the hypothalamus here. And we see our pituitary gland lying below. So that would be sitting in the sella turcica in the brain, or sorry, in the skull, right? Hanging down from the brain. They're connected by this structure called the infundibulum. So that's just this narrow bridge between the hypothalamus and the pituitary gland. When we think about its endocrine functions and talk about the pituitary gland, it's going to be very important to make sure we remember whether we're talking about the anterior lobe of the pituitary gland or the posterior lobe of the pituitary gland. You can see that it has two parts and they function extremely differently. So we will look at both, but just highlight that in your mind now as we start to think about any of these pituitary hormones that we need to know whether we're talking anterior or posterior. Okay. Posterior is easier, so we're going to talk about that first. There are only two hormones that we think about as coming from the posterior pituitary. Antidiuretic hormone, which we'll call ADH. Sometimes we'll call it vasopressin, depending on what types of effects we're thinking about. This is going to be really important for water balance and therefore for blood volume, and therefore also for blood pressure. So that will be released by the posterior pituitary, as will oxytocin. So oxytocin is particularly linked to lactation. So it'll create milk ejection. So this is also important. That's the second hormone released by our posterior pituitary. Okay. At the top here, we can see that these are called peptide hormones. This is not a thing that's really emphasized in the book or really in this course, but you should just know for yourself, for your lives, for your futures. So some hormones are going to be peptide hormones or protein-based. Other hormones are going to be steroids. So basically lipids, not protein-based. And that's important because it affects how they interact with water and therefore how they interact with fat. So in the future, you will probably pay a fair bit of attention to whether you're talking about a hormone that's soluble in water or one that's insoluble in water, because basically hormones that are not water soluble are going to stick around longer. They get into fat, stuff like that. Same thing when you think about vitamins in the future. We do want to think about water soluble versus fat soluble, really whenever we're trying to think about things that are happening in our body. So not a focus of this course, but that's why it's on the slide. That's why it's specified. Okay, so ADH and oxytocin are coming from the posterior pituitary. And part of the reason I say this is easier than what's going on in the anterior pituitary is not just that there are only two of them, it's also because of the way they get released. So we think of these as being neurohormones, and we'll sometimes refer to the posterior pituitary itself as the neurohypophysis, because the way the posterior pituitary works is basically we have neurons, their cell bodies are up here, right, in the hypothalamus. Their axons extend into the posterior pituitary, where they release their neurotransmitters. So the way in which ADH and oxytocin are released is just, we have an action potential, come down this axon, so down this neuron itself. And the neuron itself releases ADH or oxytocin. That's pretty simple, and they release it directly into the blood. So the neuron itself is releasing this hormone, essentially. Here is a short slide about what they do. We can talk about this later. What I really want us to just get down is the contrast between how the posterior pituitary versus the anterior pituitary works. So in the anterior pituitary, we'll see that the structure is different immediately. So when we were looking at the posterior pituitary, the neurons, right, the neurosecretory cells just reached from their cell bodies in the hypothalamus to sending out their hormones in the blood in the posterior pituitary. But when we're talking about the anterior pituitary, we're going to see that we have a series of steps, basically. We're going to have cells, neurosecretory cells in the hypothalamus that release neurotransmitters into the blood, and specifically into a capillary bed right here in the infundibulum. That capillary bed leads via a vein, a portal vein, into another capillary bed, which is then in contact with the cells of the anterior pituitary, which then release their own hormones. So this is a series of kind of complicated steps. So we call this system, this blood system, we'll see this set up in a couple of places in the human body. We call it a portal system. So when we have an artery leading to a capillary bed, usually elsewhere in the body, an artery will split. It'll just have like many capillary beds that are all kind of their own terminal endpoints. So this setup where we have a capillary bed feeding into a vein, feeding into another capillary bed in series, so like in order, so like any molecule of blood or any red blood cell, let's say, would travel down the artery into capillary bed 1, through the portal vein into capillary bed 2. That's not normal. It happens in only a couple places in the body, and here in the anterior pituitary is one. Okay. So what we're going to see when we're thinking about the anterior pituitary is that we're going to have to think both about the hormones that are being released by the hypothalamus here into this first capillary bed, and then also about the hormones being released by the anterior pituitary itself. So we're going to refer to this as axes, basically. We're going to be talking about, we'll talk about the name specific ones in a second, but we're going to name both the hormone that's released by the hypothalamus and the hormone that's released in response by the anterior pituitary as like kind of a group. So we have this word that describes the fact that some hormones, their job is to tell other cells to release hormones. So we call them tropic hormones. And that's what's happening here with these cells in the hypothalamus. So these neurosecretory cells in the hypothalamus, they're releasing hormones into this first capillary bed, that travel into the secondary capillary bed and tell the endocrine cells in the anterior pituitary, hey, go release your hormones. So those hormones from the hypothalamus are tropic hormones. They can be stimulating or inhibiting, but their job is either like tell the anterior pituitary to release this, stop the anterior pituitary from releasing this. So this portal system in between communicates that signal from the hypothalamus using these tropic hormones to the endocrine cells of the anterior pituitary. Well, we'll pick up back there on Friday and we'll talk about some of these specifics. Okay. Take care, everybody.